So in this last video, we're going to be talking about the cytoskeleton. These are images from the Cell Image Library website, and you can go onto that website and you can click on this image and you can color in different parts of the cell, like the nucleus, the mitochondria. And these three images are coloring the different cytoskeletal filaments that we're going to talk about in this mini lecture. But first, let me start with an overview of the cytoskeleton. The cytoskeleton is a dense, dynamic, and complex network of protein fibers that help a cell maintain its shape. So the cytoskeleton gives a cell its round shape, or if it's spread out on a substrate, and it can also help that cell resist forces like being pulled apart or compression forces like being squished together. The cytoskeleton also plays a very important role in cell movement. This can be movement occurring inside the cell, so moving organelles or vesicles from point A to point B, or it could be movement of the entire cell, like extracellular movement, including crawling. And then finally, the cytoskeleton is involved in all kinds of biological processes that we'll study this quarter, including cell division, cell contraction, and the interaction between cells. So there are three main filaments that make up the cytoskeleton, and we're going to talk about each of these. But those filaments are actin filaments, which are also called microfilaments because they're the smallest, intermediate filaments, or microtubules. Okay, so here's what actin filaments look like. Actin is primarily gathered around the periphery of the cell, although it's not depicted in this picture. You can also find actin in the center. Intermediate filaments are dispersed throughout the cell, as are microtubules, but what you'll notice about microtubules is they organize themselves and they originate from this one site that we'll call the microtubule organizing centers, okay? And this one site exists in animal cells and fungal cells, but plant cells have a lot of these. Okay, so let's talk about the individual fibers. Okay, so the first one we'll talk about are the actin filaments, and these are the smallest. Now, actin makes up around 1 to 5% of all protein inside a cell, in a generic cell, but in muscle cells, there's a lot of actin. So this shoots up to 20% of the muscle cell's protein is just actin. And actin is composed of these monomer proteins that we call G-actin, standing for globular actin. And you can see that actin is an asymmetrical protein. It has what I'm representing in this cartoon, this rounded end, and then this curved end that is bound to ATP. And when we think of actin, we can think of this globular actin as kind of loose bricks, okay? And these loose bricks can be polymerized into a brick road, okay? And so when globular actin associates with other actin monomers, right, we call this polymerization. And they're associating via non-covalent interactions, okay? Once we have one strand, this will wrap around another strand, and it will give us an actin filament. And these are the smallest, as I mentioned, but in diameter, they're about 6 nanometers. ATP hydrolysis, so you can see this ATP is going to ADP as it's being added to the growing strand, is what powers polymerization, so it provides the energy for actin to grow. And because actin is an asymmetrical protein, when these fibers form, we end up with an asymmetrical strand where we have a minus end, that's what we call it, and a plus end. And the plus end doesn't refer to charge. What it refers to is that the strand is more likely to grow or will grow more quickly at this end, the plus end, than it will at the minus end. Okay, so the rate of growth is faster at this end, but ultimately we go from bricks to a road. So actin filaments can do many things in the cell. As I mentioned, they can maintain cell shape, and they do this by resisting tension, so cells can't be pulled apart. They can also move cells via muscle contraction or cell crawling. And so you were able to either see or watch videos of amoeba crawling, and you could see them basically shoot out this projection that we call a pseudopod and move towards something, whether it was food or something, nutrients or something that, or away from light that amoeba wanted to reach. Actin also plays an important role in dividing animal cells in two. We call this cytokinesis. And it plays a role in moving organelles and cytoplasm in plants, fungi, and animals. And so again, this is something that you've already seen in the lab, either in a video or through a microscope, where you saw the chloroplasts moving in on these tracks in plant cells, and that was powered by actin filaments. And these are just some of the functions of actin. So actin helps maintain a cell shape, but I talked about some of the other functions that actin has. And many of those functions require the cooperation of motor proteins, okay? For actin, the motor protein is myosin. So when you think of actin, think of the myosin motor protein. And there's a lot of myosin in muscle, right? So muscle contraction related back to actin. This protein is able to convert chemical energy in ATP to do mechanical work. And so basically what happens is here we have an actin filament, okay? And what will happen is this motor protein has a tail region and a head. And by hydrolyzing the ATP that the motor protein is bound to, it'll basically swing this head region, okay, back this way. And so eventually it'll move, kind of like pulling a rope, or here I have a cable car, right? It'll kind of start pulling the rope towards the plus end. And so the cargo and myosin will eventually move towards the plus end. And again, as I mentioned, this is really important for movement, but also for cytokinesis and for cytoplasmic streaming in plants that you observed in the Elodea. The next group of proteins I want to talk about are intermediate filaments. These are about 10 nanometers in diameter, and they're composed of various types of proteins. So actin's always made up of actin, but an intermediate filament can be made from something like keratin or laminins, okay? There are about 70 different types of intermediate filament proteins in humans, but what they do is they assemble into these large strands that then form helical structures with each other, and then those helical structures assemble to form these bundles, and that's what helps them maintain cell shape. They do not have, because they're all kind of similar proteins, they do not have polarity, and they do not have motor proteins that interact with them, and so their primary role is to support the structure of cells. Okay, so just a few examples of specific intermediate filaments that are worth talking about. So we have intracellular keratin, and this provides mechanical strength to cells and helps them resist pressure. We have the nuclear lamins, and we'll talk a lot more about these when we talk about mitosis. They hold the nucleus in place, and they help the nucleus maintain its shape. And then secreted keratins are probably where you've heard of keratin before, and so most cytoskeletal proteins are inside the cell, but this form of keratin can be secreted, and it will then form fingernails or toes or hair, right? So, but again, most cytoskeletal filaments we're going to talk about in this class are inside the cell. So that brings me to our last group of cytoskeletal filaments, and those are microtubules. These are the largest cytoskeletal elements, and they're made up of tubulin, okay? And so microtubules are made up, tubulin is actually made up a dimer of two proteins, and those would be alpha tubulin and beta tubulin, shown here, okay? And these proteins are bound to GTP, right? So GTP is a lot like ATP, except the nitrogenous base has been substituted. Okay, so GTP stores a lot of energy and will provide the energy for microtubules to polymerize. So first we get a single strand of microtubules polymerizing, and we call that strand a protofilament. And then 13 of those protofilaments combine to form a tube. And that tube is 25 nanometers in diameter, so it's the largest. And like actin, tubulin is not symmetrical. And so we end up again with polarity on our ends, where we have a minus end and a plus end. And the plus end will grow, or polymerization will happen more quickly from this plus end. Okay. As I mentioned, microtubules, at least will arise from a microtubule organizing center. And at least in animal and fungal cells, we just have one of these. So here's a picture of what a microtubule organizing center looks like. It is made up of a centrosome, which contains a pair of centrioles. And so centrioles are made up of microtubules in this unique organization. Okay, so there's this centrosome, and then there's this amorphous material in the matrix that has proteins that can help polymerization occur. And then we have growth, where it goes from the minus end to the plus end and continues to grow at the plus end. Okay, microtubules do a lot in the cell. They provide stability by stiffening cells. They also provide a structural framework for anchoring organelles. So without microtubules, the ER and the Golgi will just turn into vesicles and kind of disappear. Microtubules play a really important role for separating chromosomes in mitosis, and we'll look a lot at this. Microtubules move organelles, and they mediate the movement of cilia and flagella, okay? And they have other functions. Microtubules work with motor proteins as well, okay? And so the two motor proteins for microtubule are kinesin and dynein. And so I just want to explain a little bit about how kinesin was discovered. So Ronald Vale and his team were working with these squid axons. Okay, so these were really large, very, very large nerve cells in the squid. And you could basically squeeze out the cytoplasm, and you could make these videos to watch microtubules polymerizing. And so here in this video, this long fiber here represents the hollow rod that would be a microtubule. And what happened was he saw that vesicles would move along this microtubule, so or organelles would move along this microtubule. But there needed to be ATP present, because there needed to be energy to move things. But if you just added vesicles and ATP and microtubules, that still wasn't enough. What was still needed was a motor protein. And so this led to him discovering kinesin and dynein. And so I'll play this video.
In this experiment, a cell homogenate containing many different organelles is added to microtubules. Motor proteins are normally attached to the organelles. When ATP is added as a fuel for the motor proteins, some organelles bind microtubules and are moved along the tracks by their motors. Most kinesin motors move towards the plus end of microtubules. Dynein motors always move in the opposite direction. Both motors are used to transport organelles, and occasionally a single organelle, which must have both types of motor attached, can be seen to switch directions. The bidirectional traffic observed here is reminiscent of that in an intact cell.
So I think that video is pretty cool. And so if we were to zoom in on what kinesin would look like, what you see is we have another tail region, and then we have a head. The head will grab onto the microtubule, and it will walk for kinesin in the plus direction, and the tail will hold on to that cargo. So just a summary of the 3 cytoskeletal filaments. We have the actin filaments, the intermediate filaments, and the microtubules. You should know what they're made of and their various functions that they play in the cell. Okay, so that's it for this video, and I look forward to seeing everyone in class. Thanks, bye.